Contract to act. A deep dive into the microscopic mechanisms of muscle contraction. What is a muscle? Muscles are organs containing specialized cells that allow for many kinds of movement. 3 types of muscle tissue: skeletal, attached to bones or to skin, cardiac, found in the walls of the heart, and smooth, found in the walls of hollow visceral organs, intrinsic eye muscles, airways, and large arteries. Cardiac and smooth muscles are under involuntary control, while skeletal muscles are under voluntary control. Regardless of a specific muscle's characteristics, it will be made up of cells called muscle fibers. The endomysium surrounds each muscle fiber. A fascicle is a bundle of muscle fibers covered by a connective tissue sheath known as the paramecium. All of the fascicles together form the entire muscle, which is surrounded by a layer of fibrous connective tissue called the epimysium. All of these connective tissue coverings fuse to form tendons, which attach muscle to bone. Upon neurosis, attach muscles to other muscles or to bone. Just beneath the sarcolemma and the sarcoplasmic reticulum, there are many nuclei and mitochondria. There's also myofibrils, which are incredibly important for muscle contraction, as they contain actin, a thin filament, and myosin, a thick filament. The unit of a muscle fiber is called a sarcomere. The striation patterns of skeletal muscle have 2 main parts: I bands, composed of thin actin filaments held together by structures called Z-lines, and the A bands, which are composed of thick myosin filaments overlapping thin actin filaments. In order for a muscle to contract, an action potential needs to be generated in the axon hillock of a motor neuron. This action potential will travel down the length of the axon until it reaches the axon terminal of the motor neuron. Where the motor neuron and the effector skeletal muscle fiber meet is called a synaptic cleft, specifically the neuromuscular junction. This entire unit is called the motor end plate. Within the axon terminal of the motor neuron, there are vesicles containing the neurotransmitter acetylcholine. When the action potential reaches the end of the axon, some of these vesicles are released into the synaptic cleft. Acetylcholine rapidly diffuses across the synaptic cleft and binds to specific acetylcholine receptors in the muscle fiber membrane. As a result, ligand-gated ion channels or chemically gated ion channels open sodium-potassium channels. In its resting state, potassium is more concentrated within the muscle fiber, and sodium is more concentrated outside. By means of active transport, sodium floods into the muscle fiber through these channels and potassium floods out. This causes what is known as depolarization to occur, therefore altering the membrane potential of the skeletal muscle fiber. The resting potential sits around -70 millivolts. With this change in ion concentration, the resting membrane potential rises to -50 millivolts. The sodium-potassium pump will maintain this membrane concentration and return it to its resting state, but not before this net movement of positive charges moves into the cell at the motor end plate opens nearby voltage-gated sodium channels on the sarcolemma. This big positive charge or action potential will travel down the transverse tubules in the sarcolemma. In the action potential travels, it will trigger the release of calcium to flow into the cytosol. When a muscle fiber is at rest, troponin and tropomyosin block the binding sites on actin molecules. The calcium released will bind to troponin, therefore changing its shape and altering the position of tropomyosin. The movement of tropomyosin from its original position will expose the binding sites on actin filaments, allowing linkages to form between myosin heads and actin, forming cross bridges. According to the sliding filament model, when sarcomeres shorten, the thick and thin filaments slide past one another, with thin filaments moving towards the center of the sarcomere. As this occurs, the H zones and the I bands narrow, the regions of overlap widen, and the Z lines move closer together, shortening the sarcomere. This is called cross-bridge cycling. The breakdown of ATP puts the myosin head in a cocked position. When a muscle is stimulated to contract, a cocked myosin head attaches to actin, forming a cross bridge that pulls the actin filament toward the center of the sarcomere. When another ATP binds, the myosin head first detaches from this actin binding site, then breaks down the ATP to return to the cocked position. The cycle repeats as long as ATP is present and action potentials release acetylcholine at the neuromuscular junction. These are the student learning outcomes listed for this section, as well as the terms needed to know, which were all covered in this presentation. For my research portion, I decided to focus on idiopathic inflammatory myopathies, or IIMs. These are also known as myositis. This is relevant to my topic as this directly correlates with microscopic muscle. The first thing I learned from this article is that IIMs are a group of autoimmune conditions in which the body attacks its own skeletal muscle tissue. As a result of these attacks, a common feature of these conditions is inflammation of skeletal muscle, therefore causing muscle weakness. I also learned that this disorder affects more than just the muscle tissue itself. Lundberg et al. Explains that the inflammation of the muscle tissue also affects other organs, including the skin, joints, lungs, gastrointestinal tract, and heart. Aside from muscle inflammation, I learned that other symptoms of this condition include skin rashes and microscopic tissue features. Based on these differing presentations, there are subsets of IIMs. Some of these are dermatomyositis, polymyositis, inclusion body myositis, and immune-mediated necrotizing myopathy. Finally, I learned how to diagnose some of these diseases. A limitation with this subgrouping is that there are overlapping symptoms with each, making it harder to pinpoint and diagnose which subgroup these patients may have. It is important that we discover more about these diseases and their classifications as to develop new therapies that are ethically superior for patients affected. The myositis-specific antibodies, autoantibodies, are helpful in supporting the diagnosis, classification, and subclassification of IIMs, but circling back to our histology unit, muscle biopsies with histopathological evaluation are still important in many cases to rule out other myopathies. These are a list of my sources.